Alright guys, so this video will cover the introductory portion of our final unit of the semester, which takes a look at histology, which is the study of human tissues, and then also gives you a little insight into what forensics is. And so if you're intrigued by that and you're a junior, you can take a look at maybe taking the forensics class next year. Or if you're a senior, you can look into forensics courses in college as well. So this is my old house, actually. I don't live here anymore, but those of you who maybe voted for my house in the past to win the decorating contest were voting for this house. I'll have to show you a picture of my new house whenever I get it decorated, because I don't really know how I'm going to do that yet. Okay, here's your essential questions, and the biggest thing here that we're trying to get at is, why do we study tissues? Why is that important? How is structure related to function? So we looked at this for cells before. Now we're going to look at this for a little bit bigger structure, a group of cells called a tissue. And then that kind of fits into why we find tissues where we find them. And it's mainly because of what they do. And then, last but not least, we're also going to talk about forensics as we move through this. So this introductory portion of the notes will kind of give you a little background on what forensic science is. And then as we move through studying different types of tissue, we're just going to kind of plug in little forensic fast facts kind of here and there to see how it applies to tissue studies. And this is like my favorite picture from my wedding. Don't tell my wife that I said that, but this is my cousin Matt, who you've seen in a couple other things so far. And I think within 5 minutes of getting to our reception, he had lost his dress shirt and just had on the vest that you see there. And then this is usually what I do with Matt, is I laugh at him. All right, so histology. You say histology and people usually think history and those sorts of things, but histology is the study of tissues of the body and the way that those tissues are kind of put together to form organs in the body. This is a very important part of medical training, and honestly one of the toughest courses that I took when I was in an undergraduate at U of L. And I think I've talked about this in class actually before, that it was a course that was pretty much primarily just in a laboratory with a microscope, and you had to study all these different tissue types with a microscope, and you were kind of on your own. Now that can be extremely challenging. But in our case, I don't think that you'll find it to be as difficult for a couple of reasons. One reason is that we're actually not going to use the microscopes at all. There's a couple of reasons for that. One is that it's a lot harder with a microscope. It's a lot harder for you and I to be looking at and seeing the same exact thing for me to teach you about it. Other reason is that we really don't have that many slide samples of human tissue to look at. In fact, we don't really have any. And our microscopes also aren't the greatest in the world. So instead of using microscopes, we're going to use pictures. Pictures that I've found that I think are kind of perfect representations of the different tissues. And so that way we can look at them together in the notes and learn them that way. The other reason that I don't think you'll find this to be terribly difficult is you're actually going to get to use the notes when we take our test over this. So in a rare kind of blast to the past, I'm going to give you a paper set of notes to go along with this. You can also always access this, the digital version on your iPad, but then on test day, you'll be able to use that paper version to help you. Now, moving forward on this slide, tissues are groups of cells that work together to do a particular function. And generally a tissue is going to be cells of the same type, then grouped with other cells that are differentiated. So different types as well, but all of them are working towards a common functional goal. And so like as we've seen this year, every tissue, just like cells, has a particular structure that fits with its function. So for instance, here's our first slide to look at here. This is actually called adipose tissue or fat tissue. What you're seeing here is some adipose cells or fat cells, which are what these white circles are. They're almost like a bubble. But the tissue itself Also contains a little bit of connective tissue, which we'll talk about more later. Here in pink, you see the dark spots are all nuclei. And then this thing right here, circled in blue, is actually a blood vessel. Now, that blood vessel is like coming out of the screen at you right now. So it's been cut off, and you're kind of looking at the end of it like a tree trunk. So you got the wall of the vessel out here, and then inside here are actually some red blood cells. So this tissue, this adipose tissue, doesn't just contain fat cells, it contains some other cells as well. Now, sorry, the pictures are messed up on this slide. It may be hard to imagine how we get from a three-dimensional organism to a two-dimensional picture like you see on the last slide. You know, those fat cells, like I said, are like bubbles, and that blood vessel, as you know, is like a tube. But when you look at it in 2 dimensions like that, you just see circles. Okay? So it's kind of difficult sometimes to do a histology study using the microscope, those sort of concepts, because you've got to be able to, in your mind, picture a 3-dimensional organ or a 3-dimensional tissue, but what it would look like as a 2-dimensional snapshot or picture like you see there. Now, scientists use kind of a basic 4-step method to fix a tissue, make the tissue, onto a slide under a microscope so that you can actually study it. Now, the first key, as we saw with our microscope lab weeks ago, is you got to have a really thin section. So basically, we want to try to take a slice of a tissue that's 1 cell thick, which is extremely hard to do. But we want to get it really thin so that the light can pass through it and we can see all those structures. So, step 1. All right, step 1 is to fix the tissue. Now, what we mean when we say fix the tissue is we're going to soak that tissue in some chemicals to stop its natural deterioration. Primarily what we're doing is we're getting rid of any bacteria, so it won't break down, it won't decompose. Basically, we're going to try to slow that deterioration that would normally happen after a cell dies or a tissue dies. Generally, you're going to use formaldehyde is the classic thing. They don't really use formaldehyde anymore for specimens like we will dissect, because formaldehyde is a little bit caustic, a little dangerous. And even when you do use formaldehyde, you use 4% formaldehyde. So most of that solution is still water. Formaldehyde straight up is a really potent and nasty chemical. But if you use it just a little bit, then it has that ability to fix the tissue. So that's the first thing we'll do. Second thing we do is we embed or mount the tissue. Now what that means is we're going to first dehydrate it. Now that doesn't mean just leave it out and let it dry. We're actually going to dehydrate it chemically. So we're going to put it into a solution that's more or less a salt solution that will suck the water out of the tissue. And then you set that tissue into a solid block. The cheapest and easiest way to do this is to put it into a block of wax or paraffin wax. So basically you put your tissue, whatever it is, if it's a brain like this picture, you put the brain into the middle of a block of wax. Or kind of the expensive way to do it, which is what a lot of companies will do now if you buy slides from them, like the slides we've looked at this year, is they will mount the tissue in plastic. So that's going to obviously hold it together maybe a little bit better, be stronger than wax, but that is a little bit more expensive option. Now, our third step, then, once we have this tissue mounted in a cube of wax, is to slice that cube. So we're going to slice that cube as thin as possible into little slices. A lot of times we call this sectioning the tissue. And you can cut that tissue at all different angles, all different directions to get different viewpoints in your sample. But again, ultimately our goal is to just get a really, really thin slice that we can then lay on a slide and be able to shine the light through. So this top part of this picture is kind of weird. It's just showing you different types of cuts you could do using an egg as an example, because that's a structure we're all familiar with. So like this top part of the picture would be showing you a longitudinal cut through the egg, and that could happen at all different layers like it's showing you here, and each one would give us a different view. The second part of the picture here would be an example of a transverse cut. So this up here was longitudinal to top, transverse cut, side to side, right? And again, we could do that at all different levels in this egg, and it would end up looking differently at each level. And then at the bottom, it's showing you more like a realistic example, right, of these cuts. So let's say we have a blood vessel like this here. A longitudinal cut, like here at A, might give us something like this. A transverse cut might give us something like this. But then you can get all kinds of other things too, right? This would also be an example of transverse, and it looks very different if you look at those two together. This would be an example of transverse, and again, it looks a little different. And then right here at C, we'd call this an oblique cut, which is not really straight across in any direction. So main important thing to take away here is look at all the different ways that this blood vessel could look if we cut it. So again, it becomes quite a challenge to kind of wrap your head around what things might look like on a slide. Now, the fourth and final step that we do, so we fix it, we mount it, we section it, and then we have to stain it. And this is because most of your tissues are colorless once the blood drains out of them. So not that you would see nothing under a microscope, but you couldn't really see much in the way of detail. So instead, we stain it. We use a dye and expose that to the tissue, and then usually you kind of rinse the dye away, and it leaves some of the tissue stained. The most common stain we see is called H&E, which stands for hematoxylin and eosin, which is a mouthful. You can definitely just say H&E. An H&E stain gives you a purple, pink, blue, red kind of color to the tissue that looks like this. You might recognize it because it's probably the stain that was used when you looked at an animal tissue back in our microscope lab before. But hematoxylin and eosin is a unique stain because it's really a combination of two things. One thing that is acidic and one thing that is basic. So as a guide here, I wrote out kind of what we see. If a tissue component absorbs an acidic dye really well, we say that part of the tissue is acidophilic, and it will give us a pink shade. So as you notice in this picture, these pink areas are cytoplasm. Cytoplasm is very acidophilic and comes back pink. But if a tissue component takes up and soaks in these basic dye better, then we say that structure is basophilic, and it dyes sort of a blue or purple color. So the darker circles that you see in this picture are nuclei, and nuclei are very basophilic. They will show up very purple and much darker usually with an H&E stain. So as we move forward, most of the slides we look at are going to have this purple and pink shade to them, and that's because they've been stained with H&E stain. But as you'll see, there are other stains also that give us different looks, and we'll see a couple of those mixed in. So why study histology? And the reason I say to study histology is because it's important in lots of different areas of science. One is research. Almost every research project that you see on a living thing at the university level will involve some histology. So no matter what the group is studying, maybe they're just studying like how the brain works or something like that, they will probably at some point in their research take a sample from a brain and look at it under a microscope and study the cells too. Second example here is medicine. Medical pathology is one option as a specialty for doctors, and a pathologist is an expert at looking through a microscope. He or she will look at a tissue and can tell you if that tissue is diseased or not. And so they obviously have a really strong background in histology, and then they learn how to find tissues that are sick. And then the third thing, which is what we're going to see as we move through this little unit, is forensic science. Tissue samples are oftentimes used as evidence when we break down a crime scene. And so that evidence can help us to potentially identify a suspect or a victim, and then potentially catch the criminal. So we're going to see how that kind of relates here as we move forward. So what is forensic science? And again, I messed up the picture on this slide too. Sorry about that. Forensic science is the science of associating people, places, and things involved in criminal activities. There's your book definition. So forensic, right, that word comes from the Latin term forum, which means public or legal, right? And then science, we already know, right? Scientific method, a technique used to understand the world. So can we apply the scientific method to matters of law or public concern? And that's what forensic science is all about. Now, crime scene investigation, CSI, has gotten lots of attention because of the television shows that were around over recent years. But CSI is actually only a piece of forensics. The actual investigation and breakdown of a crime scene is a really important part of forensics, but it's not the only part. Now, obviously, you're in science class right now, so we're not going to look at the law or legal side of this that much, but we are going to talk about the science aspects of it. Now, forensic science is broken down into several different areas. I think I've got 8 of them here in this little PowerPoint that I mentioned briefly. There's really even more than that. And so this is not, I like this because it maybe gives you some insight into what a forensic scientist would do, and maybe help you realize that there are some things that forensic scientists do that aren't what we necessarily have seen on television, but that might be a cool career for you if it interests you. So first thing's criminalistics. This is what you've seen on television. This is crime scene investigation. Those people are criminalists. So these are police officers who have been trained scientifically to break down a crime scene. And a lot of times criminalistics is what we refer to when we say forensic science, but as I've said so far here, it's not the only part of it. Now this is where you collect and analyze physical evidence. Right? So you might be picking up fingerprints like this guy in the picture, but you also might be studying drugs, weapons, tool marks, human remains, blood, other body fluids, footwear, tracks from cars, tracks from feet, all kinds of different things that would be evidence at a crime scene. Second thing is forensic pathology. Now this is the branch of forensics that's in charge of determining cause of death and manner of death, if a death occurred under suspicious or unknown circumstances. So a forensic pathologist or medical examiner is actually a doctor. Right? This is a physician who is specially trained in basically dead people, right? Clinical pathology. This is not usually a police officer. This is not somebody who's trained in on that side of things, but this is an actual doctor who went through medical school and all that kind of stuff. And their main job is to perform a post-mortem exam or autopsy to try to determine how someone died. Now, up here at the top, I've got cause of death and manner of death, which you're probably thinking that's the same thing, but it's actually different, right? Cause of death is what specifically killed the person. Manner of death is kind of how that happened. So, for instance, cause of death might be a single gunshot wound to the abdomen. Manner of death then could be homicide, if it was murder, could be suicide, if it was self-inflicted, could be accidental, if it was an accident, and things like that. So cause of death, a little more specific. Manner of death kind of becomes a group thing. How and why did the cause happen? Third thing here is forensic anthropology. This is a branch of anthropology, which is the study of humans and their ancestors. And I realized here in your notes, I don't think I changed it yet either, that this was worded poorly because it said function is to identify people who cannot be identified. Well, then why are we trying, right? So their function is to identify people of unknown identity, right? And the way we can do that is looking at their soft tissue features, looking at their fingerprints, looking at their skeletal remains like you see in this picture. These people might help with facial reconstructions if bones are found. And a forensic anthropologist will try to determine how old someone was by the size of their body, maybe their sex based on the shape of their bones, how tall they were, what race they were, their socioeconomic status. You can sometimes tell from human remains whether they were wealthy or not, and all those kind of things. Now, a forensic odontologist or forensic dentist helps to identify deceased people through dental records, also might examine bite marks as evidence to a crime, But a forensic dentist, too, is just a normal dentist. This is not their full-time job. They probably have a full-time job working as a dentist for you and me, but then maybe on the side they do some forensic odontology work with their local police department. Now, an example here on the next slide of the importance of dentistry is the Ted Bundy case. You've maybe heard of Ted Bundy before. Very creepy serial killer dude, a very famous serial killer. Ted Bundy was crafty. He was clever. He was very intelligent. He would trick women into helping him. He would wear a cast that he didn't really need, or a sling on his arm, or he'd use some crutches, and he'd go to a store or the shopping mall, and he'd be in the parking lot, and he'd make a big show of not being able to carry his groceries or whatever it might be, and these women would offer to help him, and when they did, they'd find out that he didn't really need the crutches and that his arm was fine or his leg was fine, and he'd get them into his car. Bundy eventually confessed to 30 different murders committed in the mid to late 1970s, but he's often boasted that he killed more people than that even. So the total number people aren't real sure of. These are 6 different ways that Bundy looked over the course of time. He was actually thought to be a nice-looking man, at that time, and again, he was very charming and just a kind of a scary predator for females. What's interesting is that he kind of got away with things for a long time, and then he eventually got picked up on a traffic violation. So the police caught him, I don't know exactly what it was, running a stop sign or something, and then they were able to connect him to murders by bite marks and dental evidence with remains from his victims they had found with bite marks on them. But again, going back to how intelligent he was, he actually defended himself in court. He wasn't a lawyer, but he chose not to have a lawyer, and he defended himself fairly well compared, against the evidence that they had on him. Now, another area is forensic engineering, and again, this is not somebody whose full-time job is to just look into this, but maybe a civil engineer would, on the side, work with crime scene investigators to look at failure analysis. So if a building collapses or something like that and causes some death. Also, forensic engineer might help to reconstruct a traffic accident or something like that would involve a lot of physics, just to help the crime scene investigators and the forensic specialists figure out kind of what happened. You also have what's called question documents. Again, this person probably works for a museum or something like that during the day, but then might on the side work with crime scene investigators to look at documents. Might be that they're trying to figure out if documents are forged or authentic. Might be trying to figure out what was written on something that had been erased. Might be looking at an official document to see if it's been altered. Might be looking at pieces of burned documents from a fire. All kinds of stuff like that. Looking at types of paper and ink and whether a certain document came from a particular printer. Just an expert in documents. Now the last 2 here, toxicology, right, involves chemical analysis of body fluids to see if there was a poison involved. Notice the poison that's usually involved is drugs, right? And we can also look at the content, how much of that drug might be present and what that might do to a person. Notice here I've noted that over 50% of criminal cases are alcohol related, which is just another reason not to drink, right? Messes with your decision-making process, and then that's when people commit crimes. So we don't often think of alcohol as a poison or even a drug, but it is. So realize that's often involved. And then the last one here is behavioral sciences. Here's Hannibal Lecter up here, if anybody likes The Silence of the Lambs. Forensic psychiatrists and psychologists help to determine a person's competency, also might help in interviewing suspects and create psychological profiles, so trying to understand a criminal better. In some cases, these are just everyday psychiatrists, right, who, again, might work with the general public during the day, but then help in a forensic setting on the side. or they might really be forensic psychologists. And if you're a fan of the shows like Criminal Minds and those kind of shows, profilers that you see on some of those cop shows, these are psychologists. They try to understand the criminal better. And again, certainly detectives in a police force do a lot of interviewing and interrogating. But if you have a really high-profile criminal, a serial killer, somebody like that, you might also bring in one of these psychiatrists or psychologists to help with that. Now, it all comes back to evidence, right? Evidence is the information we use in a legal investigation to either support a claim or refute a claim, right? Now, evidence can be testimony, right? It can be spoken word, it can be documents, or it can be a physical object, like you see in these pictures. And again, evidence might be there to help support what you claim, might be there to help refute what the other side of the legal investigation is claiming. But I just like evidence. I think it's cool because it's a physical representation of what we do in science, right? Everything we claim in science, every hypothesis we make, we have to support with evidence, and that's how the legal system works as well. Now, tissue samples are very commonly used as evidence in a criminal investigation, particularly when we're talking about some sort of homicide investigation or something like that. We are most certainly going to see tissues become evidence. Now, studying histology gives us a bunch of different types of evidence. For example, trace evidence, tiny stuff, right, could include hair and skin, right, which, as we'll see in a minute, are both examples of tissues. Serology, or the study of bodily fluids. We study blood, we study other fluids as well, which are actually tissues, right? And then postmortem findings. Characteristics of the body after death are going to show up at the cell level and at the tissue level as well to tell us certain things, maybe about cause of death, maybe about how long someone's been dead, or those sorts of things. Now, that shifts us back into our study of histology. And then, like I said, we'll kind of drop in some forensic facts here and there as we move through the tissues. So in class, we're going to take a look at four major, or sometimes we call them the fundamental groups of tissues. These include epithelial tissues, which are going to be at the surface of things, connective tissues, which do exactly what they sound like, right? Connect things, muscular or muscle tissue, which I think you're familiar with, and then nervous tissue that makes up our brain, spinal cord, and nerves. Now, you often have maybe heard the term epithelial before, our first group of tissues that we're going to study, if you like those crime dramas on TV, because they like to drop that word, right? They say, well, we found some epithelials at the scene. And that's very realistic. We leave our epithelial cells everywhere because epithelial cells are on the covering of something. Now, that might mean the actual covering of the outside of your body. Your skin is a great example of an epithelial, but it also means coverings inside. So the coverings on your airways or the coverings on your digestive lining, those are also epithelial tissues. But the example that we often leave behind is skin cells. Right? Skin cells die and they fall off of us all the time. I mean, that's primarily what dust is human skin cells. So if you touch stuff, if you sneeze, all sorts of things, you will leave skin cells behind that theoretically could be found. And then we also have hairs, right? Hairs that fall off of our bodies constantly, especially if you were to rub your head or something like that. Those hairs can provide evidence as well. So epithelial tissue, we'll define this real quick and then that's where we'll wrap up. An epithelial tissue is a tight group of cells that form a sheet that covers the surface of something and then lines your cavities. So it covers the surface of your skin. It also lines the inner cavities of your body. So anything that enters or leaves the actual internal portion of your body, your bloodstream, has to be absorbed through an epithelial layer. Right? It has to cross somewhere, whether it's absorbed in your lungs or through your digestive tube or through your skin. It has to go through this tissue. And that's why it's here, in part, is to give this nice, tightly connected covering that might not be so easy to get through, to get into that internal part of your body. Now, there's really three shapes of epithelial cells that we'll see on the next slide, and they can be arranged in either single layers or multiple layers. Now, the base of every epithelial tissue is called the basal lamina, or the basement membrane, like you see in this picture. So I'm going to outline it in blue here. This is the bottom of the epithelial tissue. Below that, this is connective tissue, which we'll come back to later, but above that would be your epithelials. And in this picture, it looks like maybe several layers of cells here, but we'll come back and talk about this specific example later. So when we look at epithelials, we've got 3 possible cell shapes, and we got 3 different types of layering. First cell shape, squamous cells or squamous cells. A few of you ran into this term when you looked into types of cancer. Squamous cells are really flat and thin. They have very little cytoplasm. They're just kind of like a pancake. Second thing, cuboidal is what it sounds like. Cube-like, right? Almost a square. Usually kind of has rounded edges to it, but almost a cube-shaped cell. And then the third option is called columnar. And again, it's what it sounds like, columns. These are tall, thin cells that end up shaped like columns that we see in architecture. And then we also have 3 types of layering. A simple epithelial tissue has a single layer, just a single layer of cells. A stratified epithelium has several layers of cells. And then a pseudo-stratified, pseudo meaning false, right? Pseudo-stratified looks like several layers, but it actually is only a single layer of cells, and so we'll see some examples of that as well. And here to wrap up is a picture of the 7 different epithelial types we see, which are all a combination of the cell types and layering types we saw in the last slide. So you can have simple squamous, which is a single layer of flat cells. You can have simple cuboidal, which is a single layer of cube-like cells. And you can have simple columnar, a single layer of column-shaped cells. Or you can have multiple layers, right? Stratified squamous, several layers of flat cells. Stratified cuboidal, several layers of cube-shaped cells. Pseudo-stratified columnar. You don't see true stratified columnar. They don't really stack on each other. But you can see this thing that looks like columnar and looks like multiple cells, and that's pseudo-stratified. And then the last one is a weird one that we'll look at later, which is found only one place in your body, and it's called transitional epithelium. It almost can change shape. Right? So that's kind of a cool one. Okay, so I know this was a lot of introductory material. But moving forward then in class, we're going to look at some specific tissue types. We're going to look at some pictures of those, and we'll get you ready to knock out this last unit before our Christmas break. Thanks for listening.